Hi, I'm John Chodera. I'm an associate member in the Computational and Systems Biology program at the Memorial Sloan Kettering Cancer Center, and it's my honor today to be speaking on behalf of the COVID Moonshot, an open science collaboration developing a small molecule orally bioavailable inhibitor of the SARS-CoV-2 main viral protease for COVID-19 therapy. We all know that COVID-19 is caused by a novel coronavirus, but this wasn't known until the 10th of January in 2020 when the novel coronavirus sequence was first uploaded by Chinese scientists and finding that there is a striking similarity to SARS-CoV and MERS-CoV. Not only that, but the viral genome was strikingly similar to SARS-CoV-1 in a number of particularly important places. One of those places is the main viral protease. Now, the main viral protease, or Mpro, also called NSP5 for non-structural protein 5, or 3CL-Pro for 3C-like protease, it exists as a dimer and is a cysteine protease that cleaves the polyprotein that's translated by the virus in PP1AB or PP1A in 11 different places, making it an essential protease in the life cycle of maturation of this virus. The idea is that maybe you could interfere with it with a small molecule drug and shut down the virus early in infection, preventing infection from taking hold or treating patients who first come down with symptoms. Now, Mpro is highly conserved among viruses that cause SARS, MERS, and COVID. You could see the sequence similarity is strikingly similar to a number of different beta coronaviruses. And then when the structure was solved first on 5th of February 2020 by a Chinese group, the structure bore striking resemblance to earlier Mpro structures from other beta coronaviruses. Therefore, it appears that this might be a viable target for antiviral therapy and potentially even for pan-coronavirus therapy if it is so highly conserved amongst beta coronaviruses and possibly other coronaviruses. Now, while no human coronavirus Mpro inhibitors have been approved as a drug, there is a coronavirus inhibitor known and used in treatment of at least in experimental treatments of cats. Given this success in mammals, it seems like it's possible that we could develop a new Mpro inhibitor that it could be successful in humans as a therapy. Now, the problem has been that these inhibitors, including the feline one, are difficult to develop, or at least all known inhibitors up to this point are peptidomimetic and often covalent. They have this covalent warhead, for example, in the box highlighted here. And these peptidomimetic drugs are very difficult to develop into useful oral drugs. Even Pfizer, which has brought a peptidomimetic Mpro inhibitor to the clinic, has gone through intravenous administration route. And the problem is that this can only be delivered in a hospital setting. And by the time you're going to a hospital for COVID-19, you're already so sick that the opportunity for impacting viral infection before immune reaction is already passed. So it might be too late to deliver a useful therapy. Instead, an oral therapy could be taken early after infection or after exposure, allowing us to actually shut down the mechanism of the virus. So we really needed a new potent small molecule drug that was not peptidomimetic, but the question was, how do we get there quickly if we have no starting materials to go from? Normally, drug discovery is a long process, but Diamond Light Source attempted to kickstart this process using a very exciting method of high-throughput fragment screens. Now, on February 14th, the main viral protease sequence had been transmitted, and the particular details of crystallography had been transmitted from the Chinese laboratory that had to close down to Diamond Light Source in the UK. They were able to clone and produce large quantities of protein that they were able to crystallize and determine the atomic structure of at Diamond on February 20th. They then followed that with a covalent screen and a high-throughput small molecule screen, allowing them to shoot up to 1,500 crystals in a single day, which they then solved rapidly, identifying 78 different fragment-bound structures that they immediately released to the web to help the community advance therapeutic research for coronavirus disease. This resulted in a total of 48 covalent fragments bound to that catalytic cysteine 145, and a total of 71 active site fragments, including covalent and non-covalent, and a few at the dimer. The way this works is actually really incredible. These protein crystals are grown in large high-throughput screening trays, which allows dispensing from a echo dispensing acoustic dispenser using an inverted plate. It uses acoustic droplet ejection to eject small nanoliter droplets that then soak into the crystals. If the small molecule likes to be in the crystal, then it'll immediately soak in. If it doesn't, then it'll stay unbound. And then simply by solving these crystal structures and identifying which of these have density for a ligand in the active site, we're able to figure out which ones or where whether or not the crystal actually has a small molecule within it. To do this, of course, they use a fully automated beamline where they're able to solve or to shoot 1,500 crystals in a single day using this automation system. And then by assembling them together, they were able to find a number of different fragment structures that cover the active site essentially completely. Zooming in, we can see the sites S1, S2, S3, S4. These are pockets in which the peptide, the viral peptide, binds in order to be recognized by the protease and then cleaved. And the cleavage happens at the cysteine 145, which is catalytically involved between S1 and S1 prime. You can see what was really particularly interesting is that some of these fragments appear to be, which are soluble and look like parts of drugs, appear to be binding in almost identical ways, sharing some of their chemical moieties in a way that could allow you to think about moving towards more potent inhibitors simply by creating compounds that merged the features of these two small fragments. The question was then, could these fragment merges reveal a path to going straight to potent inhibitors and skipping many of the intermediate steps that happen in normal drug discovery projects? What happened, of course, is that Diamond immediately released all of this information to the web. In fact, you could see Marty Walsh tweeting about the results. All of the data was summarized in a website. All of it was made downloadable. It was enabled to be viewed interactively through a tool called Fragalysis. And now what normally happens is that these initial hits would then go through a target to lead stage or hit to lead stage, and then a lead optimization stage, and then preclinical studies. This whole process takes several years and has a high attrition rate in normal drug discovery, and then costs a lot of money, of course, when you get to the final stage through clinical trials. It's usually extremely long and expensive, but maybe given the global pandemic nature, it was possible to shortcut some of this process. So the initial question was, what strategies would most quickly get us from these fragment structures all the way to a useful drug? And near London, at the Weizmann Institute, who had been involved in this covalent fragment screen, suggested, why not try all of them? There's lots of researchers around the world that have different methodologies for how they could utilize this information to work towards potent molecules rapidly. Why not tap all of them? And so Near tweeted this tweet here, calling all medicinal chemists, we release 60 structures of fragments bound to the Mpro SARS-CoV-2. We're crowdsourcing ideas for what to synthesize and test next. You design it and we'll test the best designs. And sure enough, the response was literally overwhelming. Laboratories around the world use different methodologies, some designing by hand, some designing by clever algorithms, some designing using docking or other ligand-based methods. There's a huge number of designs that were submitted in a very short time. I think we allowed a couple of weeks for this to happen. Overall, there were 7,000 designs submitted by over 350 designers. The first 850 compounds that were synthetically feasible and not crazy, as determined by simple docking filters, were made, and over 800 compounds were tested. We found a large number of hits in the better than micromolar range. This is really, really incredible. In order to do this in such a short amount of time and at scale, Post-Era, who had been contributing their services for a number of different parts of organizing the Moonshot, they're an AI discovery company in which one of their tools, Manifold, allows us to identify how to rapidly synthesize small molecules with high success rates, given the building blocks that are currently in stock in multiple CROs, in this case, Enamine, Wuxi, and Assay. All of these CROs were tapped to rapidly synthesize as many compounds as were possible given Post-Era's AI algorithm, which also has some published work here you can see in the corner. And the data was immediately reported back to the community. All of the affinities, and because this was done at Diamond Light Source, we were able to get structures often within a week of their compound receipt, and then posted to the web immediately. So all of the affinities and all of the structures were immediately reported for the community to digest and to advance any discovery efforts around the world. But then we thought we really have to pursue this in a way that ensures that these small molecules end up in the clinic once we get something that will actually be useful and drug-like. And making a drug is distinct from actually making a small molecule that is a tight binder. So at this point, we needed to tap a larger effort, collaboration in order to bring this to fruition. The idea that everyone embraced was that this should be done through open science, where all the data is reported and all of the decisions are made in the open. All of the data is made available on this website, postera.ai/COVID, where you can find it all right now. And this would be done without patents because of the need to rapidly pursue and make available small molecules that could treat COVID disease or other subsequent diseases at minimal cost around the world and maybe produced by multiple factories. Also, patent-free allows a collaboration like this to flourish as rapid and move as rapidly as possible without needing to sign multi-party agreements. Quickly, this became a vastly global effort, where laboratories around the world were contributing various different resources or operating at cost or contributing in kind, often assays, sometimes synthesis, in order to make this distributed drug development effort really work. Of course, you might ask, why do we need oral drugs if we have vaccines? Now, if you're vaccinating 100% of the public, which is over 7 billion people, you need absolute safety. You can't tolerate even a small degree of unsafety compared to an oral drug that's only taken by sick people for a few days. You also don't need 100% compliance by the public, which is unfortunately something that we have to worry about in the United States. Oral drugs could also be deployed very early, unlike IV drugs or antibodies, like I mentioned before. You could also potentially remain effective against mutations that are challenging the vaccine, right, as we're seeing right now, because the main viral protease is so highly conserved, unlike the surface of the spike protein. An oral inhibitor, of course, that may not require cold chain storage, would be much more easily delivered out to the third world, where you don't need cold supply chains. Currently, these vaccines, for example, require -20 or -80 Celsius refrigeration, which is quite a challenge. And finally, the oral inhibitor could provide prophylaxis following exposure or treat acute illness right after the onset of symptoms, rather than requiring something like IV administration for other potential therapeutics. So to get to this point, we tapped Ed Griffin at MedChemica, a veteran of AstraZeneca, a 20-year veteran of AstraZeneca, who then convened a panel of medicinal chemistry experts contributing their time with over 100 years of industry experience amongst them. They drafted this target product profile, which is a set of conditions that one needs to meet in order to have a good chance of having a molecule that might actually be a useful drug and perform well in clinical trials. This is sort of the first step in defining what you're actually trying to achieve, and as you'll see, it requires more than just anti-protease activity, but also needs to actually be effective against the virus when it applied to cells. It needs to have a decent enough coronavirus activity spectrum at this point. And it also needs other properties like solubility and tolerability. It has to have a good half-life under oral administration, good PK. And then there's a host of other safety issues that one has to consider in making sure that it will be safely tolerated and will pose no problems in clinical trials. So given this target product profile, you also need to design the assays that you'll use to monitor your progress towards this. And this is the assay cascade we came up with, asking everything from does it actually inhibit Mpro and how does it bind in order to help us drive those optimization cycles very rapidly on the weekly timescale, to other tiers where we ask whether it kills the virus in infected cells. And those assays often need to happen in BSL-3 laboratories because of the nature of the infective virus. And then finally, there are animal models that could potentially tell us about how well we're actually achieving in vivo efficacy. In this case, humanized ACE2 mice seem to be a useful animal model, though there's still a lot of uncertainty about whether success in those animal models correlates with clinical endpoints that the FDA would look for. So what was exciting about this effort is that this crowdsourcing process immediately generated a number of high-potency chemical lead compounds simply by merging several of the fragments in a clever way. Here's one you can see where a number of them have been merged into this 3-aminopyridine compound. You can see the 3-aminopyridines on the left side, and then there's a benzene ring with a chloride poking into the pocket P2 on the right side. This was 23 micromolar, but it was still within our measurable range of about 100 micromolar or less. So it was a good starting point from which we could actually look whether we were going in the right direction through cycles of iterative design. We got so many different lead compounds that it was impossible to follow up on all of them. All of the data is on the website, so anybody could progress these if they want, but we decided to focus on 3 different series. Actually, let me tell you, the numbering here corresponds to the S1, S2 that I showed you earlier. So P1 is on the right in this case. That's the pocket that the first amino acid side chain across the scissile bond, the peptide bond sits, and then P1 prime is up at the top. That's the other side of the scissile peptide bond where the protease cuts. And then these other P's are just reserved for the side chains of the normal peptide substrate. You can see a peptidomimetic in the upper left, the coverage of the active site in the upper right, and then we focused on 3 non-covalent lead series, the 3 aminopyridines, which span P1 and P2, but also could poke into P1 prime and P4. We also had a number of quidolones as a backup series that we explored, and then another backup series of benzotriazoles, which were actually from SARS-1 inhibitors that we started to optimize for SARS-CoV-2. So we focus entirely, and for the rest of the talk, I'll focus on this 3 aminopyridine series we explored quite broadly. Immediately, we tried things like poking up into P1 prime or P4 and optimizing the different substituents in the different pockets and finding that many of these changes could lead towards increases in potency and could also be combined in an almost linearly combinable way. In fact, you can go all the way to make this compound, which pokes into all of those sites, but is unfortunately too lipophilic and has liability in metabolism on that beta-lactam ring. That means that isn't, even though it's more potent, it isn't the direction we wanted to go in. Instead, we've spent most of our time focusing on how to optimize this P1, P2 highly ligand efficient scaffold, and have managed to get this all the way to 80 nanomolar and have determined the structure of it tightly bound in that P1, P2 pocket. This pocket is highly conserved, so we think it's less likely to elicit resistance or be sensitive to resistance, and it has sub-micromolar antiviral activity, which is very exciting. We think we might have to go a little bit further, of course, to optimize this by poking into P1 prime or P4 by adding additional substituents, but we're very happy with how far and how efficient this particular compound is. This isn't, the only part of the story, just antiviral activity. There's a lot of other checkboxes we have to check off on that TPP, and we have a good idea that these compounds on the leaderboard, for example, have got us much of the way there. We're still optimizing many of these, but at least for several of the properties, there's good quantitative structure-activity relationships that allow us to make changes and then combine them in a way that pushes us at least in the right direction. So we've got plenty of room to move in this multidimensional optimization to achieve the target product profile. There's also the exciting opportunity that we could covalentize this ligand, much to the excitement of Near London, whose lab focuses on covalent inhibitors. By simply adding this acrylamide, we're able to take the scaffold and make it 230 nanomolar and irreversible, at least in this particular half-hour incubation. There's a nice, beautiful structure of this covalently bound as well, so we are clear on the mechanism of action. And we recently, as of yesterday, have a new inhibitor in this series that brings us back down to 51 nanomolar after 30 minutes of incubation. So how can we design these optimal P1 prime and P4 scaffold substituents? Briefly, I'm going to tell you about the role of Folding at Home, which is a worldwide distributed computing system that I have been involved in, where people around the world run screensavers, or at least used to, often they're gamers, which have graphics processors, in order to contribute their simulation effort to our global project. We thought we had a great distributed computing engine that was cranking away at 100 petaflops, so we thought we might turn our efforts towards aiding all of our experimental collaborators who were working on various COVID-19 discovery projects at the time. We actually got the entire Folding at Home Consortium involved, which also include Greg Bowman's lab at Washington University and the Volts lab at Temple University. And we were all working on different aspects of this in order to aid our various experimental collaborators who were working on COVID-19. What was surprising is that after posting this initial blog post, people got wind of this, and then soon, by May, over 1 million volunteers had contributed their computing power to our fight. We ended up creating the first exaflops computing platform in the world, which had been something that the supercomputing community had been trying to do for a while. Using all of this information, or all of this compute power at our disposal, we started to do very large-scale relative free energy calculations, where we would start with a small molecule that we had a crystal structure of, and then enumerate a very large virtual library of thousands of compounds we could synthesize from building blocks available at Enamine, and then identify which of these might lead to the best increases in potency to change out a different part of our molecule or explore a different pocket. The way this works is that we start with our X-ray structure, we do constrained enumeration of poses, we select a pose that's least clashy, and then we conduct a relative alchemical free energy calculation. And as you can see on the right, the retrospective performance with the Open Force Field Initiative Parsley Force Field 1.2.0 seemed extremely good for the molecules we had already synthesized. So we put this immediately into production, and very rapidly started these sprints in which we would go through many thousands of free energy calculations. Here's just one example in which we went through 10 milliseconds of simulation time in just a couple of weeks. Just to give you an idea of what this might look like is that we went through several thousand of these for one of our earlier sprints in which we were trying to optimize P4 substituents, as you can see here on the right, and ended up picking a short list of compounds to synthesize. And of those that were synthesized, we did manage to make improvements in affinity in many of them. However, those that we chose, we hadn't learned that we should pre-filter our compounds to eliminate the ones that were too lipophilic for our purposes. So we didn't find anything that we ended up using in the actual production molecule yet. What was really interesting about these sprints is that we found how few of these designs were actually significantly better than the designs we started with. It was actually very difficult to find compounds that would progress the affinity. What was also very interesting is that we also prioritized lists of compounds that humans had come up with, as well as the entire virtual libraries that were enumerated. Humans consistently seem to be better than the virtual libraries, but there are so many compounds we could generate, compound ideas we could generate in the virtual libraries, that it seemed like we were always getting more potent hits from the computer than from the humans, simply because humans can't generate thousands of ideas. Sprint 5, the latest sprint that has just run, builds on our highly potent P1, P2 scaffold through a series of different routes the chemists had worked out to prioritize from about 15,000 different compounds we could synthesize. Again, we docked these structures, and then we also created a science dashboard that you can follow along on the leaderboard what turns out to be the most potent. In this case, there's several hundred designs that appear to be more potent than our current, at least from free energy predictions. So we're now making several of those, synthesizing those through Enamine, and we're also uploading the structures so you can inspect them and see how they correspond to both the current leads and the original fragments even, what strikingly bind in a strikingly similar way. So our goal here is to use this to help progress past the last thresholds and maintaining potency or increasing potency past our 50 nanomolar biochemical potency, and then reducing the antiviral concentration needed to get the EC50. And now we're in this late stage of lead optimization here, where we aim to nominate a clinical candidate for IND-enabling studies by April of 2021. It's going to take a lot more money after that to get to the point where we can actually go into clinical trials, but we're hoping and exploring partnerships with various entities in order to help us do that. Unfortunately, I should also say that this is really truly a global collaboration. Here's just a few of the names that were involved in this, that are involved in this collaboration. You can find the complete list of contributors, which is growing every day at this URL. So for our next steps, as I said, we're currently working to identify a partner for IND-enabling studies and select a clinical candidate by April of 2021. These will be the final preclinical studies before clinical trials, at which point we hope to get our molecule into humans. We still need funding to get there, of course. We're now burning $104,000 a month on chemistry alone, but we haven't received any NIH funding to date. Our proposal for the COVID-19 emergency R1 was simply not discussed, along with a lot of other folks. We resorted to a GoFundMe to try to raise money to pay for final chemistry. It's one of the only projects I've had which we've had to do that. But once this is into IND-enabling studies, we're immediately planning to go back to all of the rich data we have and the multiple lead series in order to follow on development of a pan-coronavirus inhibitor to hope to prevent future pandemics. We think Mpro is a great target, and by optimizing against multiple Mpros from different coronaviruses, alpha and beta, we can deliver an effective therapy that will prevent this from happening in the future. With that, I'd like to thank you for your time and thank you for your attention, and to give you a list of URLs. If you'd like to learn more, then please follow the COVID Moonshot on Twitter, check out the preprint, which shows all the assays, or check out our data. And thank you again, and happy to take questions.